Okay, good morning, team. Thank you for joining Malin and I. The purpose of our training this morning is to take a deep dive into the BIOJUVE clinical study. And feel free to ask questions along the way. If you've got your study handy, I know you're driving, Elaine, so no worries if you've got your study handy. Be a good idea to pull that out, have a highlighter pen handy so you can take some notes. This is a presentation that we did at the global sales meeting when we launched BIOJUVE. I think it was very well received by the team and was really helpful. So in using your clinical study, what we highly suggest when you're presenting the clinical study is to use those slides that are contained within the BIOJUVE sales presentation. Stick to those slides, and because it really provides a nice summary of this. If you try to use this to detail physicians, you're going to get a little lost. Although, you know, there are some good places that you can point to during this study, and we'll talk about that a little bit. And it's something as a team that Danny and I are really kind of working on developing some resources to help you guys use it as a detail aid, maybe go to certain points of it and point out. But by and large, it's really used for background information and to give you an idea of what's in this study, what it means. And in those cases, when you get questions from doctors, you can say, oh, well, that's in our clinical study. Let me show you where, doctor. You know, you'll want to focus on this part when you read the study because it'll provide the answers to your questions. It's more so used like that at this time. So why utilize the BIOJUVE clinical study? You know, for those of you who come from, do any of you come from a pharma background? No, okay. And Victoria, I don't believe you do as well. You know, so for those of us who come from the pharma background or a background like Malin, who was a nurse and then esthetician and worked in medical practices, we're all quite accustomed to presenting clinical studies because it's what you do on a regular basis. But for many of our team members, you haven't worked in that environment. So we wanted to take a little bit of a step back and try to explain the importance of having a study of this caliber. First of all, this is considered peer-reviewed published data, so it's highly regarded by physicians. This is a very credible source of information for them. When they get our fancy marketing pieces, they may like those, they may provide some information, but they consider anything that's peer-reviewed and published to be highly credible, because journals aren't going to publish information like this unless those who are editors of the journal say, yeah, this is science. And this is worth publishing. So it really adds a lot of credibility to the science of BIOJUVE. Really provides a detailed resource. Again, it tells everything of why, how, what, where we got started with this, where we took it, where our scientists took it. So this is everything from the preclinical all the way to the clinical study in subjects. So preclinical is, how did Dr. Thomas Hitchcock and team of scientists land on C. acnes defendans, what they did to put the genetic switch into C. acnes defendans to make it a proprietary strain to Crown Laboratories. And then the clinical study, when we say clinical study, that means it was done in humans, in patients, but we call them subjects. So then the clinical study in 121 subjects. So that's why this publication is 16 pages long. I will say that in working with our advisory board, which was a group of very well-known dermatologists across the US who have been guiding us over the last couple of years in bringing BIOJUVE to market and developing resources and such, that we received a lot of praise for this article. The way the study was done, the way the article is written has been highly praised. Now, how would you utilize this? And we talked about this a little bit already, but basically, you're going to share the basics of the study and leave a copy for them to review. Now, the basics, again, are already in your BIOJUVE sales presentation. That's a good way to be able to provide that information. There's also a QR code in those slides. So instead of always handing out a very expensive piece of paper, you might even just want to ask, you know, is this something of interest? Would you like to dive into the clinical study? Doctor, you know, do you prefer digital or do you prefer hard copy? Because if you prefer just a digital copy, let me share with you this QR code, scan it, it'll take you right to the resource. And that way you can save some of your printed ones. We did do a very high quality print on this clinical study. You'll see that because in the before and after pictures, they're really well done. You can actually get a good idea of the results from those before and after pictures. Before and after pictures are always going to be better on a digital screen. So really best if you were to show those from your iPad or from your laptop versus paper, but paper is always a good backup. So, understanding certain points of the study, and there's call outs in terms of your implementation guide. So y'all can see that. So really understanding, if they ask about the genetic switch, where is that in the study that they would want to read? If they ask about, does BIOJUVE have any effect on collagen? Where is that in the study? So start to learn and really dive deep into the study, not only here, but on your own time. Read it all the way through, highlight it, think about it, and be ready to present that when you do get those questions from customers. And how not to use the BIOJUVE clinical study. So of course, we are an FDA governed organization. So it's important that we follow certain guidelines when we're presenting our products and not present them in a way that would be against regulations. So in no way can we convey that BIOJUVE regimen is a drug or contains a pharmaceutical active ingredient. So you hear a lot of talk, team, about medical grade or pharmaceutical grade skincare. Those are just words, quite honestly. What we bring into the market when we sell to medical practices, we bring physician dispensed skincare. So, there is some value to, is physician dispensed skincare better? Probably so than what you can get, elsewhere. There's a lot of data behind it. It's been scrutinized by physicians. There's articles such as this, there's white papers and so forth. So yes, but not necessarily medical grade, and certainly not pharmaceutical grade. So let's be careful not to use words such as that. This is physician dispensed skin biome care. Consider that those enrolled in a study aren't patients. When we talk about a clinical study, those people who were in the clinical study are referred to as subjects, and you're going to see that throughout the study. So, really work on that. As you're presenting a clinical study, you're going to want to present it in a clinical light. You can't imply that the BIOJUVE regimen treats a medical condition. Now, you will hear this. You're going to present this information to dermatologists, and they're going to say, oh, well, I really like the reduction in erythema that you're seeing. Does this make this the right choice for my rosacea patients? The answer is, doctor, I'm going to leave that at your discretion. We are not indicated for any medical conditions. I'll leave you the 16-page article, or scan this QR code right here to get the article. You take a deep dive into the information, and you determine from your knowledge whether you feel this would be the right choice for any of those patients. If you have any questions along the way, I would say contact our medical affairs team directly at medinfo@crownaesthetics.com. They're the ones who can answer those types of questions. We can't suggest, by using this study or the protocol that's outlined in this clinical study, that every person who uses BIOJUVE is going to see these exact same results. So be careful in suggesting that. Always leave yourself a little bit of out, quite honestly, is that, the best opportunity that a patient has to achieving the same type of results that we saw, or similar results that we saw in our clinical study, is to follow the protocol. And the protocol was all 5 products. If you venture from that, then we don't really know beyond what we have in our clinical study. I'm giving you an example of what you might say to physicians. But remember, results can vary. Patients are different, their health is different, what they've been on previously is different, what treatments they've had in the last year are different. There's so many variants that it's important to understand that results can always vary. Communicate that BIOJUVE, this is something we cannot do, is communicate that BIOJUVE ingredients or formulations impart a change in the structure or function of a person's skin. So if we start getting into structure or function, then we start implying that this is a drug, and that holds us to a completely different regulatory environment that we do not want to step off into. So please ensure when you're presenting this information that you're following these guidelines. So how do you present a research article? Typically, you would start, open it up and start with the title of the article. Understand that XYCM42 is Zyprobe technology. It's the technical term in this study for Zyprobe. Those two are used interchangeably in the study. Then we would talk about after we introduce the title, we would talk about where it was published in the Journal of Cosmetic Dermatology in November of 2022. And they're going to want to know that information. And then the authors, understanding the value of having your authors and who each of these individuals are will also assist you. So this clinical study was supported financially by Crown Laboratories. You know, some might suggest, well, it's your own study. You funded it. Of course, it's going to be a little biased towards your product. Well, keep in mind, this is a peer-reviewed published article. So, you know, there are a group of editors for this journal that felt like this was science enough to publish it. So that does give us some credibility in the science. Second of all, it's not as if another company is going to fund the research of our product that leads to, you know, revenue for our product. So of course, we're going to be the ones who fund these types of studies. And then understand who each of these authors are. Mansoo Ree, Dr. Mansoo Ree is our Director of Bioscience at Crown Laboratories. He is one of the scientists that have been with Dr. Thomas Hitchcock from the beginning. So when I say the beginning, Zyprobe is the result of about 8 years of research, almost a decade of research. It is the vision and initial research of Dr. Thomas Hitchcock, who's now the Chief Science Officer of Crown Laboratories. So when Thomas and Dr. Hitchcock, was contracted with Bellis Medical, which was our original company name when we launched the SkinPen brand into the market in 2013. He was contracted with us from the beginning to act as our VP of Clinical and Medical Affairs. At the same time that he was contracted with us, he was also in his lab in La Jolla, California, conducting the research on Zyprobe, and his company name was Zyprobe Therapeutics. And so Mansoo Ree was one of the first scientists that was working with Dr. Thomas Hitchcock back in that lab in La Jolla, California. The Zyprobe Therapeutics was part of a Johnson & Johnson think tank. At the end of that research, Thomas decided not to move forward with working with Johnson & Johnson. Around that time, Crown Laboratories was acquiring Bellis Medical and also acquired Zyprobe Therapeutics and named Dr. Thomas Hitchcock as Chief Science Officer. So Mansoo Ree, thankfully, made the decision to relocate from La Jolla, California to Johnson City, Tennessee to assist with pulling through the remainder of the research. So the last 5 years of the research have been done at the Crown Laboratories facility, Crown Microbiome Institute at our headquarters in Johnson City, Tennessee. So it's a long story to tell you who Dr. Mansoo Ree is, but we're very grateful that he has come along to the journey with us at Crown Laboratories. Dr. Mona Alcom is in our Medical Affairs Department. She is VP of Clinical Affairs and Residency Training Programs. She is also one of the expert clinical graders who was involved in this particular study. She's a very valuable member of our team, not only on the SkinPen side, but also other products and helping us to conduct clinical studies. We do have a clinical study site in Dallas, Texas, and Dr. Mona Alcom is here in Dallas, Texas. Dr. Brian Jones, another important member of our team, VP of Medical Affairs, Pharmacovigilance, and the Biomedical Innovation, has been with our team a number of years, has a long history in the dermatological space with a number of different companies, brings a lot of expertise to our team, and is also an author on this paper. Dr. Sasima, and I will not attempt her last name. Unfortunately, Dr. Sasima passed while we were preparing to launch BIOJUVE, and unfortunately is no longer with us, but a very, very valuable member of our team, and we, are eternally grateful for the contribution that she made to the formulations of BIOJUVE and this particular study. Of course, Dr. Doris Day, practicing dermatologist, a longstanding relationship, if you listen to the webinars and so forth, a longstanding relationship with Dr. Thomas Hitchcock and respect between the two of them. She stuck her claim, her stake in the ground years ago, saying that she wanted to be the dermatologist in the US who was associated with the skin microbiome. So she's a big supporter of not only Dr. Thomas Hitchcock and his science, but also Zyprobe technology and BIOJUVE, and is also an author on the study. And then, of course, Dr. Thomas Hitchcock, founder of Zyprobe Therapeutics, now Chief Science Officer of Crown Laboratories. I'm going to stop there for just a second, team, and just ask if anybody has any questions so far. Okay, well, let's move on. And I will say that's a good point. You can see author contributions, kind of what each of these people did in relation to the study at the end of the study in an area called author contributions. If you ever have any questions from physicians or question yourself, you know, who conducted this particular study. Funding, of course, we talked about is from Crown Laboratories, which you would expect with this type of research. How to present the article, you know, the abstract, you're going to have estheticians, quite honestly, that are only going to read the abstract. They're not going to want or desire to dive into a 16-page research article that is as technical as this. Those may be the cases, team, that you want to have them just scan the QR code so they can save the clinical study, but don't necessarily, you know, hand out expensive reprints that we've created. Save those for those customers who really like paper copies. The younger generation typically like digital. They live in a more digital world, so really use that QR code. Again, you can find that not only in your BioJuve sales aid, the printed version, but also in your BioJuve sales presentation slide deck. So a lot of times you're just going to present the article, where it was published, the title, the abstract, kind of summarize that, which is background of why the study was conducted, the methods that were used, the results, and the final conclusion. And many times you'll just stay right there. It's just summarizing that particular area. And then there'll be times that, you know, you need to dive a little bit deeper. So what we're going to do from this point forward is really take a deep dive into the study, point out very specific areas of the study that help to fill not only some of your gaps of knowledge, but again, give you a reference point to when you get those questions from practitioners, you know exactly where to go in the study to tell them, here's where you can read more on that particular subject, doctor. So introduction. This is really when we establish that 89% of the bacteria in the hair follicles is C. acnes. So really helping practices to understand that if you're going to expect change in the skin, from a true skin probiotic, then you're going to have to use a skin native bacteria. You can't use a gut native bacteria, which is what a lot of skincare companies have done. It's really more of a marketing gimmick. They've used lactobacillus acid, which is a gut specific microbe. They put a little ingredient into their products and they claim probiotic or microbiome friendly or microbiome. They're trying to capitalize on the buzzwords and put a little something in their product that allow them to do that, but it's not a skin native bacteria and certainly not the most prevalent skin native bacteria. This is also when we link the species of C. acnes to P. acnes. If you've got any practitioners that say, wait a minute, C. acnes, I thought that, isn't that P. acnes? It was. I think it was about a decade ago that they reclassified, for whatever reason, P. acnes to C. acnes. So they're not incorrect, just help them to bridge that gap. It's just the same bacteria, just was renamed. And, you know, we'll talk a little bit about the hair follicles as well, as you've seen in your training so far. And Christy, hopefully you've seen that to your brand new, so I'm not sure how far you've gotten into it. But, you know, if you think about the skin as your largest organ, and typically that is measured at 2 meters squared on an average person. If you think about the fact that the real estate within the hair follicles and the skin appendages, such as hair follicles, if you were to pull the skin out and flatten all of those appendages, you're looking at more like 25 meters squared. So that's 10 times more than the surface of the skin is actually underneath the skin. And that's what's so exciting about Zycrobe technology, is that it's capitalizing on that particular real estate. So, you know, and that is the bulk of what the skin is what's in the hair follicle. So to have a proprietary strain of C. acnes fendens, which we call Zycrobe technology, that lives and thrives deep in those hair follicles, is of very high benefit to the patient and their skin. Now, I'm going to link this as we're going along. This graph isn't necessarily in the study, but I want to kind of bridge some gaps for you guys in terms of where else you've seen this data And so you can kind of start to put it all together in your head. So this is a particular study that's referenced in our background training on Crown Inspire for BioJuve. When we take a deep dive into the skin microbiome, this is a study that was published in 2013 that's looking at both acne patients and non-acne patients. And what you see in the yellow is how prevalent, and each line represents a different patient. So what you see in the yellow is P. acnes or C. acnes. It again wasn't reclassified at the time of the study, so it's still called P. acnes. So this is giving you proof of not only how prevalent, again, everything in gold is P. acnes, everything in other colors is a different type of bacteria that was picked up on the skin. And each of the vertical lines represents one subject in the study. So you can see how prevalent C. acnes really is across a wide range of patients. You can also see that regardless of whether that patient had acne or not, they still had a high prevalence of C. acnes. So why this is important is you will have some physicians who will say, well, wait a minute, C. acnes causes acne, right? And so your answer is, what we know now, doctor, is that you have to go down to the subspecies level to determine if a bacteria is either beneficial or pathogenic. So yes, in some cases, C. acnes is linked to acne, though I believe there are other causes beyond that, but that's one potential cause of acne. C. acnes defendants, on the other hand, that subspecies is known to be highly beneficial to the skin. So yes, they're all C. acnes, but again, they're all the same species, but different subspecies. And so this article really gives us proof that even patients who are non-acneic have a high prevalence of C. acnes in their skin. So then we start getting into the materials and the methods of this study. So we start looking at, C. acnes defendants, how they isolated that particular subspecies. I know at the time that Thomas and Munsu had landed on elements that they were looking for, or particular characteristics they were looking for in a bacteria, and they were swabbing patients looking for, or humans looking for those particular elements. And then I asked Thomas at one point, I was like, how many patients or people did you have to swab? They weren't patients because they weren't being seen clinically, and they weren't in our clinical study, so they weren't subjects, they were just humans. How many humans did you have to swab to be able to isolate or find the bacteria that you were looking for? And he said hundreds. So that gives you an idea of, here's a subspecies of bacteria that we know to be highly beneficial in the skin and on the skin. And yet Thomas and Munsu had to swab hundreds of humans to actually find it. So, that's what we talk about in the beginning of our sales presentation, is that if all is going well, then we have this really great balance of good to bad bacteria. Unfortunately, not all is going well. We're fighting genetics and aging and environment and pollutants and harsh products and harsh treatments, and you name it. Our microbiome is in chaos, which is leading to not only issues with skin health, but also issues with aging. And so that's what we are targeting when we use the BIOJUVE products and the Zycrobe technology. So you're going to see, so if anybody asks, where did the bacteria come from? This is the area that you would go to in the very first section of materials and methods. It's strain XYCM42, again, which is our proprietary strain of bacteria. The Zycrobe technology was derived from a human isolated bacteria, which we referred to as XYCW1. That's the wild type. That means that that's what was actually swabbed from the human skin. When it was modified with our genetic switch, it became proprietary to Crown Laboratories and was named XYCM42. So you're going to see reference throughout the study XYCW1, and that's the wild type. Again, that's untouched C. acnes defendants that was swabbed from this individual. And then you will see XYCM42, which contains our genetic switch. We're going to review the genetic switch. It's coming up here. So I know that was one of the questions that you had, Elaine. So I'm just about to talk. Well, I actually, as I see that it's right here. So we're moving forward with that now. So this is the area that you go to. This is right at the beginning. I think it's like the second page of the study that looks at the genetic switch. It reveals how the genetic switch was inserted into the isolated strain of C. acnes, subspecies defendants, to create the Zyprobe technology, which is a proprietary strain to Crown. And this took our scientists 3 years. This was actually one of the most notable things that our scientists did, was to find a way to insert a genetic switch that once triggered, and it's triggered by the arabinose, or the sugar that is in the activating mist and is in the conditioning cleanse. So once triggered, that signals to the Zyprobes to spend less time growing and replicating, more time outputting what we have trademarked the term bioessentials. And again, the bioessentials are what naturally the Zyprobes are producing and releasing, which are copious amounts of antioxidants, polypeptides, proteins, short chain fatty acids, and we'll get into the value of those as we move along. So we do still want the Zyprobes to grow and replicate some, because that's what allows them to adhere or engraft on and in the skin. So you want some of that, but what we really want once they engraft is for them to spend all of their energy, or the bulk of their energy, producing and releasing bioessentials. I'm going to stop there because that's a really important concept, team. Any questions or comments around that? Okay, so we start diving into the structure of the clinical study, which was an 8-week study. We started with 121 subjects that were generally in good health. 108 completed the study. You're going to see a subset of 32 of those patients that were treated in Dallas, Texas, in our clinical study site. Those were the the subjects that were evaluated via bioinstrumentation, also clinical grading of clinical photography, but also bioinstrumentation. So that's where we get the results in the graphs that we have in erythema, hydration, sebum detection, and something else. I'm drawing a blank on, I believe there's 4 of them. So we'll get to that when we get to that part of the study, but just know a subset of those were seen in Dallas at our clinical study site. 12 subjects were discontinued from the study. They withdrew for personal reasons, none of which were product-related adverse events, which is really important. So Elaine shared with you, some feedback that she'd gotten from a patient here or there, or somebody trialing the product of, well, I'm experiencing this. And we certainly want to take that serious. We want to understand more what may be going on, give them some solutions of what they could try or do in that circumstance. But we also want to suggest that in our clinical study of 121 subjects, we did not see any adverse related events. Now, of course, this is a very controlled environment. You guys are going to see these patients, had a washout period of just 3 days, but a washout period, they didn't go directly from one product line onto trying to balance their microbiome with BIOJUVE. There was a little bit of a time period there where they were washed out, is what you call in a clinical study, or given time for their skin to recalibrate prior to using other products. These were patients who had not had any procedures done within the last 12 months or during the study. So, depending on what people have done to their skin over the last 12 months could also dictate, how they're experiencing the product.
Angela, I have a question on that about like their environment of where the patients or the subjects were from. Has anybody been like, well, they were all from Dallas, so they're all living in the same type of atmosphere? What about, has anything like that come up? Or
well, that's the good news, Elaine. And there's a slide in your sales presentation that talks about that, and you'll see that in the clinical study as well. There were 4 different regions. So remember, 121 subjects, 4 different regions of the country from where those subjects were. So we had East Coast, West Coast, North, and South. And 32 of those were in the Dallas market, and those were the ones that were evaluated via bioinstrumentation in the clinical study. So it is really
okay, got it. I got a little confused.
Yep, and in that slide, it's a really good slide in your clinical study. I don't think, oh, well, here it is. Okay, so this is the slide that's actually in your BioJuve sales presentation that shows those 4 different regions, how many subjects came from each region, and that is important. You know, there's different parts of the country, there's weather conditions, there's moisture conditions and so forth. So it is true that you want to have a diverse population. Our scientists certainly thought of that. You want diverse in age groups, you want diversity in ethnicity, and that all was taken to an account in the design of the study. So again, if you get questions on this, you just, you know, let me point you to this area of the study, doctor, in terms of the methods and how the study was set up. Any other questions on that?
So just the 32 in Dallas were the ones that we got our graphs from.
Exactly, because those were measured by bioinstrumentation. So we actually had to have the equipment in the clinical study site to be able to conduct that part of the study. The rest, when you look at the other graphs, and again, I think I've worked those in here too. When you look at the other graphs that just show the different endpoints on texture, you know, fine lines, wrinkling, coarse wrinkling, pigmentation, and so forth, those results were gathered through clinical grading. That means that clinical photography was taken at each of these clinical study sites. And again, these other study sites are not Crown Laboratories owned. These are independent clinical study sites that are deemed credible in the market for conducting clinical studies. That's what they do. They're regulated to be able to conduct clinical studies. And so they all followed, you know, the same protocol in terms of taking clinical level photography, and expert clinical graders looked at that photography before and after at 1 week, 4 weeks, 8 weeks, and determined what percentage of patients did they see change in their skin for each of those endpoints. So we'll talk a little bit further about how we get there, how you know, those study results were compiled. So again, 8-week study. Well, people are going to ask, why 8 weeks? Well, you know, you try to have enough time to be able to see results, quite honestly, but you don't draw on a study indefinitely, because in this case, we're looking to bring a product to market. Again, we were already at this point, you know, six or so years in the making of BioJuve, which means a lot of time, effort, and money has been spent at that point to get us to that point. We're looking to conduct a clinical study that is highly credible in terms of science, and give people an idea of the results they can expect for BioJuve, but not prolong it so that those results are delayed. So the 4 locations were Milwaukee, Wisconsin, Harrison, New York, Los Angeles, California, and Dallas, Texas, were the 4 study sites. Generally healthy women, ages 18 to 70. Discontinued all other facial, skincare, and topical products 3 days before the start of the study. So that could be something, you know, team, when you have somebody who's having a little bit of a transient reaction. Number one, it's not uncommon when you switch skincare products of any type to have some type of transient reaction in the skin. You know, even more so potentially because we're actually doing something completely different, and we're recalibrating their skin microbiome, which again could be in chaos. So, you know, we may see a little bit more of that. Something they may be able to do is to not, you know, give themselves a break from all skincare product and then restart, just as Elaine said, just start with the night product, 1 pump, not more than 1 pump. You never need more than 1 pump. Because why? Because we've got Zycrobes. I've got Zycrobes living and thriving on my skin right now. I'm not depending on what I put on tonight to be everything. I'm depending on what I put on tonight to supplement the Zycrobes that might have not engrafted and washed off when I cleansed this morning, the ones that may have died for whatever reason. I'm just looking to supplement. So you really don't need more than 1 pump, because all you're doing is supplementing at night. So, you know, more sprays, really dilute it and have them stop there. I would suggest they not use the Biome Support Complex the next morning. You know, let their skin just get used to the live Zycrobes first, and then work their way towards the Biome Support Complex, because that is another boost of the bioessentials in a bottle, and that could be potentially overwhelming their skin as well. So keep it simple, have a little washout period, and use 1 product at a time and kind of build your way to the other products would be my recommendation, or our recommendation. These patients, we withheld any medications or OTC products that might interfere with the results of the study. So no anti-inflammatory drugs, no retinoids, no topical benzoyl peroxide, etc. So we could really get results from our products alone. And patients who had any type of cosmetic treatment like fillers, toxins, peels, etc., within 12 months of the study were excluded. So again, not necessarily for you guys to remember all of this, but to remember where to go in the study should you get questions about this. Open it up, show them that particular area, and suggest that they read further should they want to know more on that information. And y'all don't hesitate, just like Elaine did, speak up. You know, I'm going to try, I'm looking at the clock too, because again, I have a hard stop at 10 o'clock, and I am going to need to scoot out of here. So I'm going to move through kind of quickly, but do not hesitate to raise your hand or to call out should you have a question. So this defines in the clinical study what actual products were used day and night, which is exactly what we're recommending in the BIOJUVE regimen now. That's why we suggest that they use all 5 SKUs of the product that, you know, that we're suggesting is, you know, not only, and again, they had different names in the study. This was prior to us giving them brand names to take it to market. So you'll see they're named a little bit different. Prebiotic cleanser is our conditioning cleanse. XYC and ferment-based serum is our biome support complex. Moisturizer is our hydrating barrier cream. Sunscreen is our sheer SPF. We did not, we had them using a Zyprobe safe makeup remover during the study. We don't recommend that now. We didn't find that a viable product to bring to market. What we're recommending are the microfiber makeup removing cloths that you see us distribute to our territory managers to use in territory. All right, again, our prebiotic cleanser is our conditioning cleanse. Live XYC M42 gel is our living essentials serum that we use at night, and prebiotic activator is our activating mist. So little different names, same protocol that you are recommending to practices now. Yes, Christy.
So what about an eye cream? Is this all, I mean, I've been using it up kind of in and around the ocular bone. Do people stop using an eye cream and all that?
We are continuing to develop additional SKUs to support these 5 original SKUs. Christy, we started out very concise.
Okay.
We do have, you're going to see in the study, significant results in the eye area because patients are taking it up to exactly where you're suggesting, you know, right at the periocular area. And, you know, Zyprobes again are living and thriving in that area, just like it is in the other part of the skin. And we are seeing some exceptional results. If you need more moisture around the eye area, then I would suggest using the hydrating barrier cream for normal to dry skin. So what I typically do at night is put on the Living Essential serum, and then I just take the hydrating barrier and I put additional right here, just to, you know, hopefully provide me a little bit moisture there, and do the same during the day. I don't need the hydrating barrier cream during the day, but I do like it around the eye area.
Okay, thank you.
Any other questions, team? And you'll see this, you know, again, when you look at your BIOJUVE sales presentation, you've got this laid out in terms of AM and PM regimens. Keep in mind, again, this does show the Living Essential Serum, the retinol version, but we highly recommend that they use the non-retinol version unless they're just completely tied to retinol. Why, team? Because we're seeing retinol type of benefits with the Zyprobe technology. So quite honestly, we think we have something better than retinol at this point. Not to disparage retinol, because, you know, it is the most widely studied anti-aging ingredient. It's done a great job in the last 20 to 30 years to get us to where we are today. We do think Zyprobe technology is going to prove to be better over time. So that's, and we can point them to areas of the clinical study that show retinol type of effects with Zyprobe technology without the irritation and so forth that you get with retinols. And then of course, you know, in your sales presentation, you've also got the day regimen as well listed. So again, I'm just trying to show you where in some of your marketing resources, you have slides that coincide with what you see in your clinical study. So this is where we start talking about the clinical grading that was done with the photography, what type of camera was used, how it was set up, and so forth. This is really important. It just, again, standardizes and adds credibility to how you conducted the study. And that was done in all 4 sites. Clinical photography and clinical grading was done in all 4 of those sites. And we talk about exactly, you know, what they were looking for. So clinical graders, clinical grading, which is physician assessment based on photography, occurred at baseline, weeks 1, 4, and 8. And they looked at all of the endpoints that you see in the graphs that are contained within your sales presentation, which include texture, lack of clarity of the skin, even skin tone, discrete and mottled pigment, fine and coarse wrinkling, under eye dark circles and puffiness, Turgor, which is rigidity of the skin. So looking at changes in the rigidity of the skin, and then overall photo damage were the endpoints they were looking for. And again, when you look at those graphs, and here they are, when you look at those graphs, what you're looking, and you'll need to explain this, team, when you're showing these graphs, and I highly, highly, highly encourage you, if you use nothing else in the slide presentation, the BioJuve sales presentation, walk them through the study. Again, these slides are going to really lay out a nice story that you can't really lay out when you open a 16-page article with a lot of writing. Quite honestly, there might be some little things we can point to in that article, and we'll talk about that, and we'll be bringing you resources to help you with that. But by and large, you've got what you need in that sales presentation, and you need to explain to them that this is clinical grading. So what you're looking at, doctors, you're looking at in the line on the left, the kind of flesh-colored line, that's week 1. The gold line is week 4, and the dark line is week 8. And so you're looking across at each endpoint, what percentage of patients, clinical graders who were judging clinical photography, what percentage of patients did they see change in these particular areas? So for instance, texture, 84% of patients in week 1, clinical graders were seeing change in texture of their skin, 87% at week 4, and 90%, over 90% at week 8. Doctor, wouldn't you agree? Pretty substantial for a clinical line to see results that quickly, and for results to continue to grow throughout the eight-week study, wouldn't you agree? Well, let's go down the line and let's look at all of the other endpoints of the study, and the same thing can be said for every endpoint, that we saw results, a significant number of patients, varied based on the endpoint, but continue to increase in percentage of patients seeing those particular results over the course of the 8-week study. And 2 different graphs detailing all of the endpoints. And this is where we get to, Christy, the under eye dark circles and the under eye puffiness. So remarkable, right? That we don't have an eye cream. We will, and there will be some things that we tweak to the products. There's a lot that we can do with Zyprobes, and there's going to be actually implications for Zyprobes far and wide across dermatology, quite honestly. So the future is very bright. BioJuve is just the first stop for Zyprobe technology. But even within the BioJuve product lines, there's certain things that we can do in terms of adjusting how much of the biocentrids we filter out of, for instance, the biome support complex to create that product. We can go higher concentrations in certain areas. We could add ingredients that could add a higher level of moisture. We could add HA, for instance, or colloidal oatmeal to further reduce erythema of the skin. There's different things that we could do with different SKUs, and our scientists are continuing to explore that.
Angela,
yeah,
really fast, would you mind backing up 2 slides and can you just touch on the differentiators between the 2 types of pigment that the graphs are showing?
Discrete, you know what, let me get back to you on that, Victoria. That's a very good question, and I want to make sure that I say it correctly. Do you know off the top of your head, Malin, the answer to that? I know we have defined it before, but I feel like I'm going to mess it up if I do it right now.
I have an idea, but I think that we should probably verify exactly what is meant for the purpose of the study.
Okay, perfect. If you will chase that down for us,
I will,
and then send us an email later. There might be there may be several questions that we need to answer after this. So good question. Let's make sure we get you absolutely the right answer. I want to say maybe it could be in the verbiage under the slides in your sales presentation too. We might have defined it there.
Okay, I'll take a peek there too. Thanks so much.
Any other questions, team? Okay, so this is where we define the bioinstrumentation, and I get questions on this. You may get, what do you mean? How did they measure hydration? Well, let's go to area 2.3 in the clinical study and look at, the bioinstrumentation that was used. And so your doctors who conduct clinical studies will ask these questions. Those are the ones who've asked it of me. This is where you can point to in that information. Again, not expected to remember it, just expected to understand where to go to answer that question for them. And then we looked at the following parameters: transepidermal water loss, elasticity of the skin, hydration, sebum, pH, and skin color. So improvements in melanin and erythema. So here are those graphs from your sales presentation that support that part of the article. And this is looking at hydration. So again, when you start presenting these, and this is all in the verbiage in your sales presentation, highly recommend that you guys really study that verbiage under each slide in the sales presentation. But hydration of the skin, no surprise, doctor. We did see an increase in hydration and then kind of plateaued. What we see with Zyprobe technology and BIOJUVE is a strengthening of the skin's natural skin barrier. And of course, when that's the case, we're holding in our own skin's moisture, our natural moisture better. So, you know, we can see not only an increase, but just a nice stability of hydration in the skin throughout the 8-week study. Erythema, again, no surprise. If you think about the fact that Zyprobes are releasing propionic acid, and propionic acid as a short chain fatty acid does have effects, is an anti-inflammatory. You would anticipate these types of results. And you're going to see these visibly when we talk about the before and after pictures, but a remarkable decrease in erythema just in week 1 and continuing to decrease throughout the 8-week period. And this is where they're going to start asking questions about, well, then could I use it for my, you know, rosacea patients? And doctor, I'll leave that to your discretion. BIOJUVE is not indicated specifically for any medical conditions such as, you know, rosacea. I will leave that at your discretion. Should you want to have a conversation with somebody on our medical affairs team about that, please email them at medinfo@crownaesthetics.com. Sebum production. Now we've changed this in our sales presentation. We don't actually affect sebum production. What is affected and what you pick up in our before and after pictures is sebum detection. So we are looking at, you know, the fact that the Zyprobes use sebum as their food source to create energy to produce bioessentials. So that's why you see a mattifying of the skin from week 1 to week 8 and continuing to progress. And we see that visibly in our before and after pictures consistently, both on forehead and nose. And again, we're not affecting sebum production. Some of them might say, well, then are you drying out the skin? No, because remember, let's go back to the hydration graph. The hydration actually improved in the skin, you know, and plateaued. So even though we saw a decrease in sebum detection, we don't see a dryness of the skin with the products. And the reason that there is no endpoint week 1 for the nose is because we only separated out those 2 areas at week 1. We started out, we were just looking at the forehead, and then we expanded that to the nose area starting at week 1. When they were seeing, I think that we're starting to see some pretty remarkable results. They wanted to capture 2 different regions of the face. Strain isolation. This is what talks about the triggering the genetic switch. So again, arabinose, which is a very specific sugar, Zyprobe specific sugar that is within the activating mist, triggers the genetic switch, which allows for cell replication and growth to decline. It's considered growth arrested, and for the Zyprobes then to spend more time producing bioessentials, which is what we're looking for. So then somebody can't suggest, number one, this is proprietary to Crown Laboratories and it's patented. So that is what allows not other companies to go and get C. acnes defendants and claim that they're going to be able to get the same type of results because they don't have a proprietary strain, they don't have a genetic switch. They aren't putting out the level of bioessentials that we can get with our Zyprobe technology. And it actually states here that, again, they're growth arrested. So there is some cell replication and growth, albeit at a more moderate rate than wild type. That means our Zyprobes, our proprietary strain, is less growth and replication than a wild type of C. acnes defendants.
Do you know how long we will hold that patent for? Is there like a time frame on that? Do you know?
I don't think so. I think that's indefinite. I think you're probably thinking more along the lines of drugs, Victoria, how patents run out and then they're allowed to go generic. I don't think that's the case for bacteria, but you know, for something like Zyprobe technology. But I will ask that question if we could ask the question too, Malin. But I haven't heard any concerns of patent running out and others being able to replicate what we've done.
Yeah.
We get into the results area. You know, this is, you know, really exciting in terms of what Zyprobes are releasing, which are referred to as metabolites or postbiotics or bioessentials is what with the term we've trademarked. So all terms, you know, are really interchangeable. But really, this is where it goes into the antioxidant effect. So anybody wanting to understand, well, don't I need antioxidant? Don't I still need to use my topical antioxidant during the day? Well, doctor, do you? Because let's talk about what Zycrobes are doing. They're living and thriving on and in the skin and releasing copious amounts of antioxidants 24/7. So why would you feel the need to supplement? Let's talk about what we're seeing. ROX-P is the specific antioxidant that is known to be released from, and I want to say that, let me see if I put it in here. No, okay. And your training presentation, there's actually a QR code that they can scan and actually get the article that shows C. acnes defendants releasing ROX-P as an antioxidant, and that ROX-P is equivalent to 12.72 milligrams of ascorbic acid in a 24-hour period. But the exciting thing is our scientists can see there are other antioxidants present as well, and they're working to characterize those antioxidants. So it's not only ROX-P that Zycrobes are releasing, there's other antioxidants there as well. So, and it states that here in the study there, this suggests that there are multiple sources of antioxidant activity secreted by not only, you know, C. acnes defendants, but also Zycrobe technology. And team, where
So,
our, okay, go
further
ahead.
study to find more information about that smaller molecule. Sorry.
100%.
Can you hear
Oh
me?
yeah. Yes, the research is ongoing every day, team. We're just at the tip of the iceberg with Zycrobe technology and BIOJUVE, just really at the tip of the iceberg of what we're going to be, you know, bringing to market. The science they've already got, which is really exciting. I won't steal the thunder, but there's already another revision or addition to this study that they're preparing to publish that will continue just supporting and supporting and supporting what we're doing and bring more light to the subject. So it's really, really exciting.
So,
yeah.
so just to go back on the stat really quick, Angela, with the 13-ish%, the 12 point, whatever it is, that is just the ROX-P equivalent. That's not showing the other antioxidant coverage.
Exactly.
So that's just what we know that the ROX-P is putting out. So eventually it could actually be more than that, but for now, what we have studied, we know it's about that for just that portion.
Yeah, let's read, and let me just read that. I don't think that that's, okay, that's in that article, and that's in that slide in your training presentation where it talks about ROX-P and C. acnes defendants, but yes, exactly right. And that particular study, remember, that's not a Zycrobe technology study, that's a C. acnes defendant study that shows that C. acnes defendants releases ROX-P, which is equivalent to, and it's releasing it at a level of 12.72, equivalent as 12.72 milligrams of ascorbic acid in a 24-hour period. So that's not Zycrobe technology. So what our scientists are saying about Zycrobe technology is, yes, we've got the ROX-P, but we're seeing other antioxidants there as well. And they're looking to characterize those.
Gotcha, thank you.
This is where you can go. Again, we're in the results section now, 3.1. This is where you can go to point to propionic acid. So they shortened it just PA, but just know that's propionic acid. You can go there to talk about the benefits of propionic acid to the skin in terms of significant tyrosinase inhibitory activity within skin melanocytes. So this is how Zycrobe technology, one of the ways in which Zycrobe technology is affecting pigmentation, or the that you saw in the study, is because propionic acid, which is a short chain fatty acid that Zycrobes produce and release, is a natural tyrosinase inhibitor. Tyrosinase inhibition, for those of you who haven't worked in the skincare market, I'm sure Christy and you can speak to it very intelligently, but it is one of the ways, many ways, that companies really target changing discolorations of the skin. It's the final step for melanin production, so it's considered a very clean or a very effective pathway if you can stop it from melanin or overproduction of melanin at that level, that it's a really complete way to stop it. So the fact that we've got, again, Zycrobes producing and releasing a tyrosinase inhibitor 24/7 deep in the hair follicles is substantial. Christy, do you have any thoughts or comments on that, being from the skincare industry?
Well, for me, this was huge because coming from Abaji Medical, we're taught, you know, hydroquinone is the gold standard to, you know, suppress the melanin from the, you know, the tyrosinase. And a lot of, you know, feedback, you know, people hear all these horror stories about hydroquinone and can they be on it indefinitely? And, you know, the ochronosis stories, there was a lot of talk about that. So I'm very excited about going back to those particular accounts and talking about this in particular, especially here in South Florida. Is a lot of issues with hyperpigmentation and sun damage and photo damage. So this for me, I've been off now my HQ for, well, since I started. So I'm really interested to see how all of this is going to work on for me.
Yeah.
So, yeah.
And know too, there's things that we can do, you know, in terms of manipulating the Zycrobes to get more activity in terms of skin discoloration. So, you know, companies like, you know, Obagi in particular, so Obagi and Zeo, those are lines that are really targeted and kind of hang their hat on skin and targeting skin discoloration, and do so by a number of different means, including hydroquinone, which is a very harsh product for the skin. You're actually supposed to give yourself breaks of hydroquinone. So, you know, we're not at all suggesting we're going to be able to, you know, do with Zycrobe technology. We don't know yet, and maybe it will be a different product that we bring to light that will assist with that and more compete. But, you know, we're not necessarily saying we're going to get the same results as Zeo or Obagi that really hang their hat and get very, very, very aggressive on skin pigmentation. But it is nice to see that we've got something with Zycrobe technology that it not only promotes skin health and changes the visible signs of aging, which your fellow Floridians can use, right? But something that is not harsh on the skin over time as well.
Yeah.
So, you know, in those cases, Christy, you've got somebody who's really stuck on those products, like, yeah, I'm not moving away from my Obagi. It's controlled my melasma or my hyperpigmentation for the first time ever. Great, but are you taking breaks from that? Let BioJuve be your break. What a great opportunity it is to have in between, or find a way to work BioJuve in. You know, we went to a, sorry, I'm watching the time team. We went to a dermatology practice, and one of the dermatologists like all over BioJuve. And this is Jessica Long and I in Scottsdale. And the next one was like, oh, I'm not moving away from my retinols. But you come to find out, well, not all patients are on retinol because they can't all tolerate it. And those who are on it aren't on it every night. So it's like, okay, so can we have your patients who aren't on retinols? And can we have you using BioJuve every other night, or evening serum every other night? Can we own that nighttime routine in between when they're using retinols? And then you start working your way in. So you find those little gaps in those types of patients where you can start to insert BioJuve. Any questions or comments on that, team? Team, I'm watching the clock closely, and I truly do have to run out in 15 minutes. If we don't finish today, then we'll circle back and do the second half, quite honestly. We'll just have to have 2 recordings, Malin, but let's see what we can get accomplished. Don't stop your questions, because your questions are wonderful, and I want you guys to get everything out of this that you need. So this is exciting. This is when we start talking about the ferment. The ferment is what is used in biome support complex. So anytime you see XYCM42 ferment mentioned, think biome support complex. This is the data that supports that particular product, and there's a lot of good data on it. So that's where we're picking up again the high levels of propionic acid. So as you can see, induces genes involved in skin barrier and proliferation, and an improved barrier is associated with improved skin hydration, reduced transepidermal water loss, so less loss of the body's and the skin's natural moisture, and improved ability to resist external stressors. And this is the graph in the training presentation that supports propionic acid and as a short chain fatty acid as one of the 3 main metabolites of microbiome fermentation. Lowers the pH of the skin, provides antioxidant and anti-inflammatory benefits, precipitates cell turnover, and acts as a tyrosinase inhibitor to reduce hyperpigmentation. So remember too, you've got some patients who are not, you know, really excited about moving away from exfoliants. They think they've been sold on the idea they need to exfoliate their skin. This is where you can suggest that propionic acid, based on the research, is a natural cell turnover agent. And that's something, again, that the Zyprobes are releasing 24/7. And I think you can point back to even before and after pictures. There's a glow to the skin, there's a health to the skin, all kind of, you know, pointing to the idea of what we're achieving with Zyprobe technology and BIOJUVE. This goes into the gene regulation studies. Again, XYCM42 or Zyprobe cells and ferment induced gene regulation of inflammation. So if they start asking, well, how do the Zyprobes, you know, reduce erythema to that extent? Okay, let's go to this part of the, you know, the study that talks about inflammation regulation in terms of not only Zyprobe, the living technology, but also the fermentation that's within, or the ferment that's within the biome support complex. And this decrease in inflammatory gene obviously promotes lessening of redness, reduction of extracellular protein degradation, and lessening of stress on the skin. Again, in the clinical results area, so now we're looking at 2-month clinical evaluation of the subpopulation. This is the 32 that were evaluated via bioinstrumentation in our Dallas office, looking at those very specific endpoints. And this is figure 10, which talks about each of those endpoints. There's a little more information in here than there is in the graphs in our sales presentation. So this may be one good area to kind of look at and digest and understand. And again, what we're looking at next is being able to give you some pointers about where to point to. If you're going to open the study, it's like what graphs to point to and what verbiage to use with that graph to make some of the points that we would like you making in sales discussions. So look for something like that to come out shortly, but this may be one area that you want to dive into in your own and kind of, you know, take a look at that and develop a talk track around that while we work to get you something out to you. Most significant improvements were skin texture, evenness of skin tone, under eye dark circles and puffiness, laxity of the skin, which we can see in the before and after pictures that we show, pigmentation, and wrinkles. In terms of overall endpoints of the study, very strong. This is where it states in the study that we did not have any patients discontinued due to adverse events, nor did we see product-related adverse events in the clinical study. So the regimen is very well tolerated. Again, what we can't account for are the nuances of patients. Anytime you go from 121 subjects to thousands nationwide, different parts of the country, different scenarios, less control and so forth, then you may see some of the things that we talked about earlier. And we're looking at ways of giving you guys the talking points to be able to address that. Now we move into the discussion section, really reinforces why C. acnes is a true skin probiotic, again, helping to separate ourselves from anybody using microbiome-friendly lactobacillus acid talk, a plant-derived cells, suggesting that they're going to change the microbiome. If they're not skin native, then it's not a true skin probiotic. And we're suggesting that's who we are. So we're really staking that claim as XYCM42, or as Zyprobe technology, being a true skin probiotic. So you get any questions on that? Oh, well, I'm using something that says microbiome. What? Let me point you to this particular area of the study, doctor, to help you understand how Zyprobe technology is a true skin native probiotic. Facultative anaerobe. This is really important to understand. That's what Zyprobes are. They're facultative anaerobes. That means they live and thrive. They love to live and thrive without oxygen deep in the hair follicles, but they can also survive on the surface of the skin in the face of oxygen. And why is that? Because they're producing copious amounts of antioxidants. So you think about, why do Zyprobes produce what they do? Why do they produce our trademark term bioessentials? It's because each of them are protected. They're building a little house around themselves to protect themselves. And all these wonderful substances they're emitting is for their own good, to live and survive and thrive. They just happen to be substances that are highly beneficial to our skin health and to change the visible signs of aging as well. So that's why they're considered facultative anaerobes, is because they can live and survive not only in the skin, but on the skin. Really important, thing to point out each time. So when your doctor suggests, well, don't I still need to use my antioxidant during the day? I don't know, do you, doctor? Because now we've got something living and thriving on and in the skin 24/7 that's releasing antioxidants. So now we're not worried about stability in the face of light and air. Now we're not worried about penetration. So and for vitamin C in particular, if it's an antioxidant vitamin C, it's water soluble and it's meant just to sit on the skin and fight free radicals and fight environmental stressors, if you will. If it's lipid based, if it's oil based vitamin C, then it's meant to hopefully penetrate the skin, cross the lipid barrier, and have some effect on collagen. So, you know, again, why would you need, we're covering both of those, and we've got evidence in our clinical study that we're covering both of those areas. So why would you feel like you need a topical C on top of BioJuve? And this is also, yeah.
Question kind of with that. So if there's any accounts that are just purchasing, you know, the hero products, I do have one account, they just wanted to get with the hero product, and I honestly wasn't just like educated enough at that time. I was just like, yep, okay, sounds good, get something. And if they're just doing the hero products, where do you suggest that they put it in their regimen? Like what step? Or is that something we really can't touch on since it wasn't studied?
Yeah, if they're just going to do the hero products, that's fine. What we say is, you know, 8 hours of biome care a day is better than none, right? So if I'm taking gut probiotics, well, every other day is better than none, right? So it's really just allowing their skin to have some level of biome care. The anecdotal data that we're getting, Elaine, you know, again, the only thing we can suggest is we did a clinical study and we proved this. So I can't really definitively tell you beyond that what to anticipate. Anecdotally, we're getting great feedback for those people who are just giving us their nighttime routine, but still using their Zeo 7 products, you know, during the day. They're still seeing significant change in their skin because, again, what we don't know is when they get up in the morning, like how quickly those products, whatever products they put on, are going to affect the microbes and to what extent, and we never will know. So there's an unknown right there, but anecdotally, what they're saying to us is, and these are some of our KOLs, like, I'm seeing change in my skin. That's why Dr. Doris Day is incorporating the Living Essential Serum and a couple of other products into her regimen, into her own skincare line. It's why Dr. Germain is saying BioJuve makes everything better. It's Leslie Fletcher on the West Coast, one of the biggest Zeo users on the West Coast, is using BioJuve personally at night. She's using it in patients and switching them completely. She personally just gave us her nighttime regimen. She goes, I feel like my skin is tighter, you know, that I've switched to BioJuve at night. So some level of biome care, what we're hearing anecdotally, is better than none.
Okay, so just as if they incorporated it at nighttime, right before, and then the biome support complex maybe right after they wash their face.
Ideally, there's 3 levels. And then, team, I'm sorry, I'm looking at the time. I'm going to have to cut off from here. So we'll have to come back and do the rest of this study. But we're looking at 3 different levels. So if you want, doctor, to have, you know, the greatest opportunity to have the results that we saw in this clinical study, then the only way we can suggest that you would get those results is 5 SKUs, because that's what we used, you know. But how exciting is that? It's just such a concise regimen. When you venture from that, we don't know. We can suggest, you know, that you're going to see different things. Anecdotally, we're hearing other things, but it's the only way we can, you know, definitively say you have an opportunity to get these same results. Now, if second best would be, and we're actually developing a visual to help y'all have this conversation. It's in the works right now. Second best would be, absolutely, you have to use our night serum. We have to get live microbes in and on your skin. And if you could just cleanse and prep your skin with conditioning cleanse, you know, before you put the microbes on, you know, activate them, obviously. Next morning, use conditioning cleanse, and then put on biome support complex. If you could give us those 3 SKUs, you know, it's a duo product, so really it's 4 products, but it's 3 SKUs. If you give us those 3 SKUs, then what you do is biome support complex has a film former in it. So there's an ingredient that helps to seal off the microbes and protect them. So as long as you're using either that or a hydrating barrier cream during the day, now we've got some level of protection for your microbes. So you can move on and use other products and makeup and so forth. And we have some assurance that we're keeping microbes living and thriving and emitting for and working for you. You know, that would be second best. But let's say, you know, that's fun, Angela, that's really cute, but I'm still not giving you my day routine. Okay, can I have your night routine? And if it's, I can't have it every night, can I have it every other night? Is there a way that we can find a place to insert living essential serum, so you can start benefiting from the idea of having something living and thriving on it in your skin and providing benefit 24/7. Unfortunately, in your case, you're just going to have to accept the fact that when you put on other products on top of it, namely preservatives in those products, what do preservatives do? They kill bacteria. So why do we know BioJuve all works together well? Because we've tested that each of our products, we use a certain preservative and we use it at levels that we assure that we're keeping the microbes living and thriving. So other lines, all bets are off. Most likely they're using not only actives or other ingredients that are harmful to the microbes, but also certainly preservative systems. Questions or comments on that, team?
Not on that, but before you stop recording, just for the sake of being within the same segment, since we're going to have two, I did get the answer on the pigmentation. And mottled pigmentation is patchy or irregular pigmentation, while discrete are isolated areas of defined pigmentation like a liver spot or a freckle.
Okay, very good. Thank you, Malin.
Yeah.
Thank you.
Thank you. That was
Team,
super
I'm
helpful.
So very sorry that I have to stop at this point. I'm at 37 and I have 48 slides. We'll reconvene. Sorry, this was kind of an unexpected circumstance today, but we'll reconvene to do the rest of this. Circle back if you've got any questions, you digest this. I really highly recommend you still go through the study and just read it, highlight it, understand it. I think you heard a really great testimonial from Jessica Long that it's her education of herself that's really driving the success she's having with BioJuve, and that's really the standard at this point. Is you just got to bring your knowledge level to a very high level. And we're all here to help you.
Thanks for the time, Angela.
Thanks,
We
team.
so appreciate it.
Okay. Thank you so much.
Have a great day.
Y'all too. Bye.
Bye-bye.